Oh, that's asking me a question. I didn't realize it was on that screen.
Recording in progress.
Sorry. All right. All right. So if you guys didn't see the announcement that I posted, the lab exam average was for everybody 77. For this class, it was 78. So good job, like genuinely good job for you guys. Because when I tell you, like when I say on there, like some people think I'm exaggerating for like dramatic effect. I am not. When I tell you that the average over years of me teaching this, and even Dr. B before me, it was worse with him. But the average for me was around 60. And I was like doing everything I could to try to, figure out, because I didn't want to water down the content any more than it already was. And how was I going to, help people learn the material better? Well, apparently what you guys needed was that study sheet, which is great. I'm not making you another one, just to be clear, because you guys know why, right? You need to learn to make your own. And that really is what this is about. If we can get you guys to make study sheets that you can learn off of, make notes like that, that can help you that much, that's what you need going forward. Learning microbiology is great, but if I can get you guys to where you can take skills that can actually help you in future classes and programs and everything, that's really what would be like the ultimate, win for me. So, that's why I'm doing what I do to try to help you guys with making notes, whether it is, get this out of here, whether it is, taking notes while I'm lecturing, or prepping beforehand, like what I went over. If you guys watched what I went over on Friday morning, I started out with, the usual, this is what people usually do crappy on these chapters. And then I started making notes over a class that I'd never taken before, genuinely, literally have never taken anatomy or physiology, ever. Never had to. I was a biochem major. You don't need that crap for biochem. You need chemistry. So why would I take it? I wasn't going into medicine. So I had never taken that before. So I went onto Dr. Morford's page, because if I was going to watch a video of somebody lecturing, it was going to be Dr. Morford and not Dr. Shearer. Don't tell him I said that. And so anyways, I figured I'd go through the notes associated with that and make like outline notes first. And then eventually I would go and watch his video. Now that's what I was going to plan on doing for my next thing on Friday. I'm not going to be doing that because, hey, we're doing chapter 15 and 16. And these are immunology. Like this is immunology. This is what we're getting for immunology. This is all the explanation we're going to get for immunology. Chapter 17, if you don't know chapters 15 and 16 well, you're going to suck at 17 because it relies on understanding immunology. So chapter 18 is easy, like no joke, like it's easy. You'll see what I mean. Okay. But it's just using basically all the stuff we've learned about to create testing procedures. Some of it you've even heard of, like ELISA and PCR. So that's not news. But 15, 16, and 17 can be a challenge, especially 15 and 16. Most people don't know how the immune system works or have a very limited understanding of how it works. So, so I figured I would go over, after doing my things people miss the most on 15 and 16, instead of watching a video and taking notes on Dr. Morford's lecture, I'm going to be, I made my little prep notes, right? That's what I did originally. But now I'm going to be putting together a rundown basically of the immune response. So I'm going to make like a chart, if you will. I'll start with like how it would start and then the breakdown of the things that come after it. And and just following through what would happen if we saw something foreign and what the response would look like and the cells that would be involved and why and how they're doing everything and whatever. So that's what I'm going to do after, the usual on Friday. So if you're interested in joining in on that or watching the recording, of course, I'm going to record it and post it just like I always do. Yeah, because chapters 15 and 16, getting into that immune system, man. Also after today, in case you aren't keeping up, we did chapter 11 over microbial control, like cleaning and things like that, sterilizing. Chapter 12, we did antibiotics. Now chapter 15, we're doing the innate immune system. Chapter 16, we will be doing the acquired immune system. 17 will be like immune problems, like autoimmune diseases, rejecting organs, all of that sort of stuff where the immune system goes wrong, allergies. And then chapter 18 is going to be the diagnostics. But yeah, in case you can't tell from me saying all that stuff, this is halfway. We're halfway to the exam right here. It came up that fast. I'm just pointing it out so that we're all on the same page, because if y'all haven't started like prepping your study materials, like I keep saying that you should, you're just going to find yourself in the same boat as always, drowning in the information, trying to study at the last minute, because next week is it. Like that's the end of the lectures next week. And then of course we have presentations over your last presentation, and then we will have the exam. To be clear, whatever disease you chose on that disease selection thing for your personal disease choice, which if you haven't done that, you're going to be in big trouble. Like, I'm not kidding. Because if you chose a skin-based disease, hey, day one, you're presenting on unit 4. Okay. You will present with your body system where your disease is from. So when it says that your personal, presentation is due by that for day 1 of unit 4, you should have it ready and ready to present. Everybody will be presenting in person, and you will be up here talking about your thing. I absolutely am going to grade you if you just read the slides and the cards. Absolutely, I will. Yeah, we're not going to rely on our teammates this time. It's just all you. So it can be worse or better depending on what your situation was with your group. Because if you lean pretty heavily on your group, now it's all on you, and you've got to show me that you learned about your disease. You are expected to present on your disease at the level that I would present on it. Your lecture on your disease is going to replace mine. So if I have to touch up on information, if it's something strange, of course, that like, I didn't explicitly, say, oh, be sure you mention this, then don't, if you're not going to get in trouble for that. But if it's basic stuff that I have to go back and touch on because you didn't cover it, you're probably going to know that you're not going to get a full score, because you're expected to cover everything that assignment asks for. I shouldn't have to go back and touch up on information that you covered for the class, because I literally am not going to lecture over that disease if you chose that disease. Some people choose obscure ones, like, I don't know, ones that wouldn't normally be covered, and that's fine. That just means that's extra stuff that you guys get to hear about. If it's not on the study sheet, you don't have to actually learn it. So there's that. But yeah, so we'll start with that. And then each chapter, yes, is associated with a system. It's like the skin, and then it's nervous, and then it is circulatory and lymph, and then it is the respiratory, and then it is GI, and then the urogenital stuff, which is STIs mostly. So that's what we'll be looking at. So whatever disease you chose, you'll be presenting on that chapter day. Okay.
It's like the nerve system of chickens. It doesn't matter that it's chickens. It's going to be the nerve
on the nervous system. Yes. Yeah. Because it still applies to how it would work. Right. But that's the idea. Yeah. Some of those ones that I don't know much about. Yeah. And if you guys I'll make a list of what days you guys are going to be presenting when we get a little closer. I promise I will. But I'll post that on the announcements so that everybody can see like what day they're presenting. So you're not going to be surprised or something.
Specific animal day for things that are only for animals.
Yeah.
Okay.
No. But yes. So yeah, I'll have that there so you guys will know what days you're presenting. Some of these guys kind of fall across different categories. So I might put you in like a chapter where you might not feel like you really felt like you were in respiratory. You thought you were in more cardio. Or you have some good argument for wanting to go on whatever day. That's, come talk to me about it. I don't care that much. Okay. So, but yeah, if it's just because you want to delay it, that's probably not going to get a win vote from me. So, but yeah, so that's what to expect with that. If you guys are, because you probably are starting to work on that at the same time as you're doing your group stuff, that's great. But it's the same kind of, it has the stuff that you need to present on in that assignment. Like it literally says, cover these things. As long as you cover those things and you can keep it brief, that's even better. So, so cover the things and keep it brief and try not to rely too much on your note cards, as you should like be doing anyways in your groups as well. So, but yeah, anybody have any questions? We're probably in lab not going to go over the lab exam, just because, like I said, people did well on it overall anyways. And whatever, people ended up missing, you probably have an idea. It's probably just things that you're like, oh, I put that, or, oh, yeah, okay, yeah, I know that that was wrong or whatever, because you were stressed and all this. If there's anything in particular that you are worried about, some sort of question that you are concerned about or whatever, come to me and we'll try to figure it out together. But other than that, I'm just going to assume that you guys kind of know. It's hard with that one since we can't go back and look at it on our computers, I'm aware. Most of that stuff you're not going to see it again. So the stains might come back, but it's not necessarily going to be the same questions. It'll be the same slides, but not necessarily the same questions. So
is there any particular question that did tend to trip people up more than?
Yeah, the endospore stain one, which if you guys didn't know, hey, there was an endospore stain on that exam. And it was the one that was in that back corner that was like closest by where I sit. And there was like, most people got the capsule stain, right? With the background and the halos and all that. But the one next to it, which was like red cells, and there was like what people kept saying were blue, little like marks and whatever in there, but those were endospores. They're teal. And that's that teal green from the malachite green, which is your primary stain. So that's what that stain was. And a lot of people were missing that one. They thought it was the gram stain. So, but after having seen, you knew that the first two stations were gram stains. I don't know how that got confused, but that is what happened. So it is what it is. But I only have so many endospore stain slides. That one actually I felt like was a very good slide. So it's just a matter of just making sure you're familiar with what it could look like and all of that. So I'm not saying everybody missed that, but that was the one that I would say. That one, and of course, okay, the obvious one, right? The obvious one that a lot of people were missing. What caused, like, how do you get tinea or whatever it is? Like, how do you get that infection? A lot of people were missing that one. Tinea is the traditional tapeworm. So you get it from undercooked meat. So that was that one. So a lot of people missed that one. I mean, that's not surprising, right? That's just one of those ones that a lot of people didn't study well on and whatever. So those two. I'm trying to think if there's anything else. Anything anybody was like, worried about? Yes.
You said that a lot of people on the study slides that a lot of people miss, like the PGLO plasmid one. Did we?
No, so that wasn't. So I think I took that question out because I was just sick of dealing with it. Yeah, the one about like the plasmid being, because that used to be on there. It was a plasma, what kind of macromolecule is it? And everybody was putting protein, and it's a DNA. It's a plasmid is DNA, and so I think I took that one out.
Literally, like that's very in the book.
Well, yes, it's in the book, and I'm saying it a lot to you guys, but like I guess back then, like I thought it was obvious as well, and so I wasn't like emphasizing it quite as much, and so people were just assuming that it was protein because we talk about the products being proteins, and I guess that's what they were assuming it was. But
I didn't know there was going to be so much color stuff on there, like what was the primary stain for this and what was now I didn't get to study. I have a nursing exam that same day, so I'm working same week. Yeah. But anyways, that's my own problem. But now I know. I'm like, okay, I need to focus more on the colors.
Yeah, so we'll see similar questions again on the final. Again, some of them will be the same, some of them will be different. Like we'll look at it from the different side or whatever. But it's not all going to be exactly the same question from the exact same slides, is what my point is. And we will also have, so that's it pretty much that we're going to see. I believe that's everything that we'll see from the first half of the class repeating on to the final. Other than that, the final is mostly new stuff. And we'll talk about what we're going to go over the rest of this semester in the lab, because it also, I point out to you guys, hey, unit 3, we're halfway through unit 3. But also, if you're not keeping track, we're about a month away from the end of the semester. 5 weeks. Can you believe that? So that's what we've got left. And there's a, what is it called? Thanksgiving in the middle there. In which I give everybody a week off, because why wouldn't you want a week off for Thanksgiving? I hate that Thanksgiving is like only like those 2 days. And I'm like, yeah, people want to go visit their family and shit. Like, I don't know. My family doesn't live in the same place as me. So yeah,
All the other ones
give the whole week. Yeah.
Even like the main campus at OSU gives a full week.
Yeah.
I don't know why this one's different.
We have a lot of questions like as faculty, definitely. Anyways, so whatever, but that's a lot. What I'm saying is that there's a lot that's going to come up, and we'll go over what we're going to go over in lab and everything too. But it's going to be over before you know it, and maybe that's a good thing, but also it just means a lot's about to happen sooner than you think. So let's go on with this. We'll talk about the lab stuff when we get in there. Today's going to be genuinely a short day in lab. It's not going to be like you come in there and I talk at you and we're gone, but it should be very quick. Everybody's been leaving like 30 minutes early. So, and there was no pre-lab. If you're freaking out, there wasn't. You didn't miss it. Okay, so because I wanted this to be an easy day back to lab so we could talk about what we're doing and not have to worry about like doing like suddenly this new stuff, just jumping right into it right after we had the exam and everything. Let's just ease our way in instead. So we're going to get into immunology. This chapter talks about the innate immune response, and then the next one will be the acquired. If you look at the arrow and the circle, you can see that the innate has quite a lot more things under it. That's going to be our first line and our second line of defense. But basically, this is going to work like a military attack on a bad guy. I'm going to walk you through it. And I know that Dr. Shearer uses a pretty similar analogy, but I have a lot more soldiers in my military. Okay, so get ready. They have more roles, but we'll walk through that and become familiar with how that stuff works. And I've got more BS before we actually start working, which is that people ask me about my other dog. You guys have met Meatball and you've seen pictures of spaghetti. This is Ripley. So she's old now, she's 12 years old, but she still runs around and plays with the boys, just like she's as young as ever. So that's Ripley. And then the motorcycle that I got, I don't know if you guys, I told you guys I was getting one, right? I think I told you. I told most of my classes. Okay, that's all right. I told my husband kept saying how he wants a motorcycle. So I sent him messages up there in the corner. I was like, that motorcycle rider's course. Maybe we should go do this. And he even like put the heart on it and whatever. Yeah, we should go do that. And so he came home one day to this. Now I'm not gonna lie, I haven't told you guys about my home and like where I live, but did I tell you guys the Goblin Core thing? No. My house, like if you've heard, if you know what I mean by Goblin Core, it's Goblin Core. It's like witchy, like dark, like cottage core, but dark, like witchy. So this is like this is my time, right? Yeah, Halloween is my time. We got married on Halloween. And so this is a year-round type of it. This is always up year-round. And so there's a lot of Halloween stuff in my home, but we have skulls and stuff everywhere, and our walls are painted black and stuff. But anyways, that's maybe that can't be that surprising. So anyways, I couldn't fit anniversary in like the areas I put verse, but happy early verse, you're gonna need those lessons. And then he was like, oh my gosh, what do you mean you're gonna get a motorcycle? I was like, yeah, but I had to finance it, so I didn't want like
Right.
I didn't want to be like, hey, now we have a monthly payment. Luckily motorcycles aren't super expensive, so we should be able to pay off pretty quickly. But anyways, that's the motorcycle that I picked out. It's gray. It's a Kawasaki Eliminator, which is pretty base, like learners, motorcycle. So, but the reason I'm telling you about this is because, guys, I went and that is my car. You guys probably see it in the parking lot. It's the bright blue Jeep. That's mine. So that you know that I definitely live loud like all of the time. Yes, so the motorcycle. So I was riding it around. I know how to ride a motorcycle and balance it and all that. But the deal is I had to learn how to shift. Mm-hmm. Motorcycles are manual transmissions. And I know how to drive a manual car. And it's the same concept. It's just applying, like your clutch is where your hand is. And then you shift with your foot. And then you have a brake at your foot and at your hand, like one's for the back and one's for the front, right? And so I'm like telling him about, like I had driven it around the little street or whatever. I'm like, yeah, okay, this is going to be fine. And I let him have the motorcycle. I'm like, hey, be sure, you're always using your clutch. And the guy that sold it to us and drove it over there, because we don't have a license, neither of us do. He's like, be sure, your clutch is your best friend. And I'm like, yeah, remember what Doug said? He said the clutch is your best friend. So if you freak out, be sure, don't just pull on your brake, pull on your clutch too, or you're going to kill your engine. And he's like, okay, whatever. It's not, if you guys have ever driven a manual transmission car and you kill the engine, it like does this jerking thing whenever it does it. So the motorcycle does too, which is way more terrifying because yes, a motorcycle, right? Because you're just balancing. So he wasn't going very fast or anything, but each time that he would kill the engine, it would do that jerk thing. And like people are apt to do in a bicycle, when you're like riding and you freak out on a bicycle and you just like jump off and you like throw it off to the side. Y'all, he dropped this motorcycle 5 times on the concrete and brand new. And I'm just like, it's okay, honey. Like, you gotta learn how to ride the motorcycle. It's great. It's fine. It's fine. It's fine. Like freak it out and whatever. So he was, being a man and whatever, trying to like do it. Like he just had to do it like a few times just to prove that he could do this. And he's like, I'm going to pull it up in the driveway. It's not a super high incline, but it is an incline, right? So you have to have some gas going. And he was more or less, once he was finally going and not dropping it every 10 seconds, mostly just like coasting in neutral. Okay. So he's ready to go up and he just couldn't get it. And he tried to walk it up it was just too heavy for him to walk up. Right. So I'm like, he keeps rolling it back. He's like, I'm going to get it. And I said, get off the motorcycle and I'm going to drive it up for you. Like, I just couldn't take it anymore. It's like, we're done. But it's good. We're going to get there. He just needs to practice. And I need to probably maybe wait for him to take the writer's course first, because it's stressing me out. Because just trying to go, because we're not trying to drive across the city before our motorcycle, class. It's just, we wanted to at least have a handle on how to operate it. But I don't know if I can help him with that. So anyways, that's my story. Probably way too long. Doesn't need to be that long, but there it is. Yeah, I'm not very patient. And especially when I'm like, I get paid to be patient with you guys. I'm not paid to be patient with my husband. So I'm not very nice. All our neighbors were out there like looking, and I'm just trying to be pleasant. And yeah. Anyway, so immunology, obviously, this is the part of your body, your system. It's a whole system in your body for surveilling things, making sure we're getting rid of the stuff that's foreign, recognizing it as foreign, and then breaking it down and destroying it as needed. So this includes anything foreign or any like parts of dead cells and tissue and stuff like that. So the way that we can tell the difference between self and foreign is going to be by certain patterns on the surface of bad guy cells, okay? Things that are repeatable patterns that we're going to be able to recognize. So we can see those as being non-self, and we have to train our bodies essentially to recognize things as self. And that's going to happen early on in the maturation process of a lot of our white cells, their ability to only distinguish between non-self and self. This is way more important with the lymphocytes, which are in the next chapter, but it still does play a part here. And we already learned about antigens, which are anything that your immune system can respond to. So these patterns that I'm talking about that are repeated patterns, they could technically be antigens. Antigens tend to be more specific than that, though. So it's just going to be like the spike protein of COVID or something like that, as opposed to peptidoglycan that all bacteria have. But it could act either way. Of course, if you can't tell the difference between self and non-self, then it's going to lead to autoimmune disorders. Autoimmune disorders, literally your immune system is not seeing you as self anymore. It's seeing you as foreign and is continuing to attack you and cause damage to the tissues in your body. Of course, it's going to include things like rheumatoid arthritis and lupus. We'll talk about that in chapter 17. That's really kind of what 17 is about. All right, so one of these, the big biggie term for the things that the innate immune system is going to recognize, these are the PAMPs, pathogen-associated molecular patterns. So they're patterns of molecules that are associated with pathogens. So obvious things like peptidoglycan. You shouldn't have peptidoglycan floating around in your body, right? That should be confined into your gut, and they have a whole system of dealing with the guys in your gut. So it shouldn't see that. We shouldn't see LPS or endotoxin, gram-negative stuff. And we shouldn't see double-stranded RNA, for example. We don't have double-stranded RNA, but viruses can have that as their genome. So that's an automatic like flag of not self. So those are obvious patterns that aren't specific to a pathogen, but our innate cells, they're not looking for a specific pathogen. They're looking for bad. This is bad and this is not self. So that's our PAMPs. They do have these receptors that they bind to that you guys don't need to, I'm not going to ask you about them. All right, some tissues and organs and stuff that are going to be playing a part in immunity. We're going to talk about the MPS here in a little bit, so I don't need to go into too much detail right now, but part of some stuff that helps your immune system function is the MPS, the mononuclear phagocyte system. It's like a highway for the cells. Then we have the extracellular fluid. It's just fluid that's filling in the spaces in the body. And the interstitium is any of those spaces that are going to be filled with that fluid in that area. The bloodstream, of course, is going to be transporting things around for us. The lymphatic system is going to be working as well. They are separate systems, but they work very close to one another and rely on, well, the lymphatic system definitely relies on the bloodstream, but yeah. And then capillaries and crap like that. The lymphatic system is basically going to be dealing with the return of extracellular fluid into the circulatory system. What do I mean by returning extracellular fluid to the circulatory system? What does that mean? It means like you had an injury of some kind, whether it broke the skin or not, doesn't really matter. Whether it introduced pathogens or not, doesn't really matter. But if you had swelling in that area, that swelling is due to fluid that went into that area. And then now you have to get that fluid back into the bloodstream. It doesn't just like absorb into the cells. It absorbs back into the bloodstream, but it doesn't go there directly. It has to go through the lymphatic system first. And there's a reason for that. And it is smart. We're going to talk about how that works, but it is the drain off system, and you go through the whole lymphatic system before you rejoin the circulatory system. So lymph fluid, you have, stepped on a nail or a tack or something, doesn't matter. You have a break in your defenses, bacteria are getting in, and you have, an injury. And now we're going to have a response to that. So our inflammatory response, our innate response, it all has to start going to that area and responding to that area and all that. When cells that are involved in that response need to get to that area, whenever we have things like, fibrinogen and stuff that's going to make clots and stuff like that, that comes with the fluid. So cells come with fluid, and then we also just need the stuff that's in the fluid itself. So that extracellular fluid goes to that area where you have your injury. And so the way that the cells that are getting into that area and the way the fluid is getting into that area is by squeezing through the cells of your capillaries. So in those thin spots where your capillaries are just like one layer of cells thick, they can squeeze between them and get into the tissue spaces. So they're not going to stay confined into the bloodstream. They can move out into the tissue itself. It's pretty cool stuff. And you know this because if you've ever cut yourself, especially when you were a kid, or if you have kids, this, then, I guess you could have this as an adult, but most people are good about cleaning their wounds when they're adults. You've ever had pus, in a wound or something like that. That's, your body fighting off the infection. It's not the infection itself. Typically it's your neutrophils that are making up most of the pus. So that cells had to get there somehow. This is how they squeeze out through the capillaries of the red blood cells, the bloodstream itself. But then we're going to have to get back in through the capillaries of the lymphatic system. So it's going to be the same sort of movement, but into the lymphatic system. We don't go back into the bloodstream. Not yet. Lymph moves only in one direction. We already said you're coming in from the bloodstream. You go to that site where you stepped on the nail, and that came from the blood. So now you got your fluid and your cells out there, but now it needs to get back out. So how's it going to do that? Well, it gets in through the lymphatic system, same way that it got out of the bloodstream, but into the lymphatic system this time, because we're going to join in with the lymph. And the lymph is going to get filtered by your lymphatic tissues, like your lymph nodes, and get checked like if you're going through a station, so you're going from country to country in Europe, and they're going to need to check your passport and stamp it every time. That's sort of like going through your lymph nodes on the way back to the circulatory system. Yes. Yes, so we're going to get to why the swollen gets happening, but that is the start of it. Yeah, so something foreign would come through that area. It is the start of it. So, so we have our guys that are sort of sentries that are stamping passports as things are going through. You want to be sure everything is going through they're supposed to be. It's also only going to be powered by your skeletal system. This is why if you have surgery, if you're able to ambulate after surgery, they want you to start walking around as soon as possible. That's not just to get your blood flowing, it's part that, but the blood flows and the lymph flows. And then we can get that whole thing circulating and acting the way that it's supposed to, get the filtration process going, start getting the proper, healing and immune response that we're supposed to. If you can't get up and ambulate, they'll put those things that squeeze your legs that somebody in my last class told me it has a name. I don't know what it is, but those,
SCDs?
Yes, yes. And they just squeeze pressure on your legs to help pump the lymph. That's what it is, to keep edema from happening. But edema is lymph. It's built up blood. It's that. So if you guys think of edema as being like fluid that's building up in a person's limbs or whatever from not moving enough, it is that. That's what lymph is, though. It's just the liquid part of your blood. It's what came with the cells when they went into an area. But you can't get it out unless you move. Okay, so this is my little terrible picture of that. But we've got our heart. And as we're coming out of the circulatory system, we have our cells and the fluid. That's what the wavy lines is, the fluid, and our cells with these little dots coming out of the bloodstream itself. But when they're ready to rejoin the rest of the body, they're going to have to go through the lymphatic vessels to get back to the circulatory system. Then they can rejoin. And that goes through lymph nodes and all of that. If you've seen these pictures, it's pretty wild. If you look at like comparing these two, the circulatory system versus the lymphatic system, they don't necessarily always line up, but at some points they do, right? Of course they do. And it makes sense. But look at how much of that, the lymphatic system there is. And we don't really think about that because you can't really see it as easily when you do dissections and things like that. These guys are a lot more fragile and frail, and they just aren't as easily to visualize as our larger arteries and stuff might be. But they're there, guarantee you. And then this picture on with the arrow is just showing you guys the circulatory process of it all. All right. Our lymphoid organs, we have primary and secondary lymphoid organs. The primary is basically where our cells are going to be made and mature. So that's going to be your bone marrow, and then also the thymus, which is like a second, it's like in your chest, it's a second part of T cell development. Talk about that in the next one. But and then the secondary lymphatic organs are going to be the lymph nodes, the spleen, and the ALTs, associated lymphatic tissues. They work like lymph nodes, but they're not in lymph node form. They're just tissues that have the same function as that. It includes your tonsils, for example. That makes sense, right? So all of your cells, all blood cells, red or white, come from your bone marrow. Cool. They go through a process called hematopoiesis, where you have the stem cell that's going to be turning into whatever blood cell that you need it to be. B lymphocytes, when we start talking about in the next chapter, the lymphocytes, B lymphocytes are going to complete their maturation in the actual bone marrow, whereas T lymphocytes are going to complete their maturation in the thymus. Cool. And it's like, yes, it says organ in your chest area, and it is like your upper chest region. And it just shrinks the older that you get, because most of your development of your immune system is going to happen when you're young. And you can't react to new things very well as you get older. So the lymph nodes themselves, they say they are small, encapsulated, bean-shaped organs. It says bean-shaped. I want to be very clear about this. They're bean-shaped. They are not bean-sized. They are much smaller than that. They're about the size of a pea. In my last, even smaller, probably half a pea, depending on who you're dealing with, but not my last job, but I used to work at the transplant immunology lab at Integris. And so we did all the testing that'll come in through for like organs and making sure that they weren't going to reject and whatever. So we would get lymph nodes in from the donors, we say brain dead, right? Typically is what is going on with them. Those donors. And so the donors' lymph nodes would come in with, And there are these tiny little things that we would have to like flush out with all this liquid to try to get the cells out so we could do the cross matching and make sure there's going to be no rejection. So they're way small, which should put it into perspective when we talk about like how, like when you get sick and your lymph nodes are swelling and you can feel them like big, right? They're like this size, you know, they're pretty big, like grape size, if not larger, that they start out that tiny, like half of a pea size. That's pretty crazy. And why is that going to happen? Well, we'll talk about it. But okay, so these guys are lymph nodes filter out. Like I said, they're the guys at the checkpoints stamping the passports, making sure everybody going through is supposed to be there. If they're not supposed to be there, no worries. As you're coming through, they're getting your passport stamped. We got plenty of guys lined up back here with guns in their hands. So that's basically what's going on. We've got all of our white cells kind of acting as sentries, just chilling, just waiting for the right time. And yeah, and that's when it is the right time, then they multiply themselves. And that's what causes your lymph nodes to grow. We see a bad guy come through and he matches up with one of those guys because again, this is going to be a matching up situation, the antigen antibody kind of matching. Very specific, but if we match up, that cell that matches it is going to just multiply really quickly. And then that expansion is what makes your lymph nodes expand. So it'll be B cells and T cells always. We always have everybody getting along in there. But as they're growing, it causes your lymph nodes to swell up because we've got all these new cells growing. It's pretty crazy how fast that can happen and how well it works. If you get swollen lymph nodes all over, obviously that's going to be indicative of systemic illness. But if you have just individual or localized, like you have a focal infection, like just parts like your sinuses and your lungs may be infected, then you're going to have just local ones involved. The spleen is a lymphoid organ as well. It's not its primary purpose. The spleen, if you guys don't know, is for filtering out, you know, dead or dying red blood cells, damaged red blood cells, stuff like that. But it also filters out in the blood itself any bad guys that could be there. Now, we just talked about filtering out in the lymph. That's where most of that filtering is going to happen. But just as a safeguard, we can filter out in the blood as well. And that's going to be thanks to the spleen. Macrophages, which we're going to learn about, are usually the ones that are going to be residing there waiting for all this to happen and checking out, making sure that everybody's supposed to be in the bloodstream and that we don't have any invaders. So spleen does the blood. And then we have the associated lymphoid tissues, salt, malt, and galt. Salt is skin associated, malt is mucosa, and then the G is the gut. And these are just like Peyer's patches and things like this. These are just going to be tissue that is going to work very similarly to the lymph nodes, but it's just diffuse tissue. It's not defined typically. The tonsils are kind of something different. They are defined tissues, but they're not true lymph nodes, but they are associated with the lymphoid tissues. All right, this is the difference between serum and plasma. Plasma comes from when you're drawing tubes, right? The plasma is the unclotted blood. Serum comes from clotted blood. If you are working in a hospital or at a vet clinic or anything like that where you're drawing tubes, the red top tubes or the tiger top tubes, which have the little wax in the bottom, it's the tiger top ones, but those guys don't have anything added to them except the wax. But then when you let them clot, we let them sit out on the countertop in the lab. And then when it's clotted, we will spin them in a centrifuge and it separates the clot from the liquid. The liquid that you get from that is serum. And so the serum is what is like serology, what you're going to be dealing with in like immunology stuff. So serology, immunology, serum. Okay. Plasma, it has stuff in it to keep it from clotting. The liquid portion of that is called plasma. I don't know. This area that's pointed to with the buffy coat, it doesn't quite look like that if you spun your tube properly, geez. But it looks like a little fuzzy white layer in there. And that is where your like lymphocytes and stuff would be. So if you're working with lymphocytes and stuff like that, you don't want it to clot. So it just depends on what you're doing. But yeah, let me tell you. So we already talked about hematopoiesis and stem cells. So let's get into the white blood cells. Another term for white blood cells, leukocytes. I'm not going to call them that a whole ton. I usually just call them white blood cells or WBCs or something like that. But they are leukocytes. Okay, we have two different versions of these, the granulocytes and the agranulocytes. And it's not anything crazy. It's based entirely off of looking at blood sample with a stain on it, like in a microscope. We take blood, we stain it, look at it in the microscope. Does it have granule looking things, little dots, little granules all in it? Granulocyte. If it doesn't, a granulocyte without granules. Okay, that's it. That's it. That's the difference. But they also happen to have drastically different roles in your body, and it's because of those granules. So the granules tend to be signaling molecules. And so the cells that are doing more of that signaling are going to have the granules. And that is just so happens to be the phils, neutrophils, basophils, eosinophils, and then also mast cells, which we're not going to talk a whole lot about. But mast cells will just be chilling out in like tissues and stuff like that, sort of monitoring all the time. Sort of like if you had like, scouts just always looking around with like their binoculars, looking for danger in the war zone area and whatever. They aren't going to fight anything, but they're going to signal to the other guys that there needs to be a fight. So mast cells aren't going to be actually doing a whole lot. They're not going to phagocytose, not mast cells. They're just going to release signals, and that's what their granules are. So, and you'll see why they have to do with, we're going to see them a lot in chapter 17 in type 1 hypersensitivities, dealing with allergies, and you'll see how that is later. But A granulocytes, these are our lymphocytes, and our monocytes and their derivatives. So it's really just two things, honestly. I know it's a list, but it's really just two things, because your lymphocytes are your B cells and your T cells. That's inclusive, okay? And then the monocytes and the derivatives. Monocytes are just kind of basic, what do I say about this? They're phagocytes, but they're going to be going around in the bloodstream. And once they get called by those other guys to go into the tissues, once they go into the tissues, they become macrophages or dendritic cells. So they're the same. They're just like differentiated by going to the tissue. Okay. These guys are going to move around between tissues thanks to that mononuclear phagocyte system. Like I said, it's like a highway of like fibers and things like that between tissues, between organs, between cells, to allow our white blood cells to go do their job around all the tissue. So it's this tissue that interconnects everybody. Monocytes, so they're the ones that are going to give rise to the macrophages and the dendritic cell. They're called mono because they have one big old giant nucleus. Okay. And so they can differentiate in the tissues and become things like alveolar macrophages, which give away where they are. But alveoli, the sacs in your lungs where we have the gas exchange happening, these macrophages are going to hang out in those areas and just kind of wait for things that you're breathing in, which is great. You should have something there. That's an entry site for a lot of things. So they hang out there and wait for bad things to get in that way. Those are the cells, by the way, that tuberculosis wants to live inside of. Starting to get crazy, right? All right. Cytokines is a fancy word for our signaling molecules. Cytokines are going to be either pro-inflammatory, that means inducing inflammation, or they're going to reduce inflammation, or they're going to be increasing or decreasing blood flow, our vasodilators or vasoconstrictors. So vaso being our blood flow. And then the growth factor cytokines that are going to help the lymphocyte growth and their response, because this is how this is going to work. We're going to step on the nail. The scout that we had with the binoculars, the mast cell, let's say, is there and sees the nail and the bacteria. And it's like, oh my God, no, 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 no. And then calls out, just releases the granules, calls out neutrophils. Let's say they're usually the first ones there. The neutrophils come and just start eating up the bad guys. And they're just like, guys, we can't contain all of this. We need to be sure that we have our support coming. We need to call the bombers. That's what we need to do. Well, they need to call the bombers. We need to go somebody to go tell the bombers what is happening and where and why. We don't want to call them into the wrong area or by accident. Like that's going to be serious business. Just like in real life, it would be serious business. And that's true with your immune system as well, because some of your immune cells literally kill your cells on purpose. So we want to be sure that we're not starting that on accident. So we're going to call in the bombers, but it takes a while for them to get everything together. In the meantime, we've got the neutrophils, the Marines. They're shooting everybody down, doing what they can to take care of that. Finally, we've got the big guns coming in, the tanks and the bombers with our B cells and our T cells. Bombers, of course, are going to be our B cells. T cells are going to be our tanks. And there's a reason, not just because of the letters, that I choose those for each other. But yeah, it takes a while for that stuff to get there.
So is that
the cytokine storm with COVID that they talk about. but a different way
Exactly.
over nutso and like, okay.
Yes, exactly that. So so when we have our cytokines, they are calling for basically your innate immune response for inflammation and everything like that. And inflammation can be damaging to your tissues. We're trying to fight off bad guys as best we can until the bombers and the tanks get here. And if you have too much of that going on, then that can cause a shock in your system. And you literally, like, your body can't handle like the vasodilation or the vasoconstriction and places where that shouldn't be happening and all of that. And it can kill you if it's bad enough. So our examples of some of these cytokines that are going to be involved in this stuff, anything interleukins. These interleukins are IL. You've probably heard of like IL something, IL-2 or IL-12 or something like this, because we use this to treat other diseases to have our immune system respond the way that we want it to while we're treating them. Same thing with like tumor necrosis factor, or TNF. We have TNF alpha and TNF beta involved in the inflammation cycle as well. You'll see down here we have serotonin and histamine. Serotonin is going to be made by platelets and intestinal cells and target our cells in our nervous system. And that makes sense. We know about that with serotonin and having to deal with your nervous response to things. But histamine is going to eventually affect your blood vessels. Obviously, that's going to be, giving you that vasodilation or more blood into like your eyes, your red eyes, your itchy eyes. Sensory nerves and neutrophils are going to get called in by histamine as well. These are things that we probably didn't realize that they were cytokines. Cytokine is just a word for our cell signals. That's it. They're usually proteins, but they signal our other cells to do things. So we're going to talk about our physical barriers and our chemical ones. We're going to go through them pretty quick because I think that this is pretty basic stuff. If I rip your skin off of you, let's say I deglove your hand. That's not only gross, but you're going to be susceptible to infection, right? So your skin is helping you keep from getting infected. Part of that is done by our waterproofing protein keratin. Yes, it's the same keratin that's in your hair, but it's just, that's protective as well. Your hair follicles and your hair shaft and your follicles themselves falling out, that can be protective. We also have, they say follicle cells are being desquamated, so we shed those a lot. That's helping get rid of microbes. Skin glands, we have sweat and oils that are going to help flush out these guys. Mucous membranes, we have the moisture that's going to flush out, guys, the blinking to remove things physically. We have the tears that are going to help wash things out, and they also have chemicals in them that are going to help with that as well. But yeah, so you definitely have to have something like that if you don't have that keratinized layer and you're moist, because bacteria like that area. Respiratory tract, we have your nasal hairs, and then we have your ciliated epithelium. We have the little cilia in your lungs. They're going to constantly be pushing out mucus. So when you have an illness or something and you're coughing a lot, it's because that mucus is getting pushed out by your cilia constantly. The genitourinary tract, where we have trickling of urine throughout the system, not just whenever you're urinating out your urethra, but in your ureters from the transfer of urine into your bladder. Then we have vaginal secretions, also providing a self-cleansing aspect in the vagina. The resident microbiota, taking up all the stadium seats, like we've said before, they can affect the pH in that area, as well as taking up any nutrients that those bad guys could want as well. And taking up just the seats. They can't sit, literally can't find a spot. So we want that. My nose is itchy now because I talked about it. So let's talk about the chemical defenses. One of the biggest one of the terms, along with keratin being our waterproofing protein, lysozyme is it's an enzyme that we find in tears and saliva, and it hydrolyzes peptidoglycans. We know what peptidoglycan is, makes up our cell walls of our bacteria. But hydrolyze, we learned about our enzymes that can go, like our hydrolases, and the ligases were the opposites of them. Remember, we know that DNA ligase brought things together, made bonds. So this was the opposite of it. So we're breaking bonds in peptidoglycan, okay? Cool stuff. We also have lactic acid and stuff in our skin and all this. Obviously, stomach has acid in it. We also have bile that's going to be digestive. Semen actually has antimicrobial chemicals, and the vagina itself has a acidic pH that's maintained by the normal microbiome. And this is just my little summary of all of those first line of defenses. That's not what we're really here for. And that's not what they all do necessarily on in their normal daily functions, but they do have these effects as well, in addition to what they're normally doing. So phagocytosis, we've mentioned this. We know that it is a thing. We know that it's something that our cells are going to need to do in order to fight off bad guys. Eating the bad guys, right? That's what phagocytosis is. We're going to eat up those bad guys. So phagocytes are going to surveil, then they're going to ingest, they're going to break it down, and they're going to help our other cells recognize their specific antigens. Because what a lot of them are going to do, so some of them are just going to go through and eat them and digest them and poop it out, like whatever, throw it up, however you want to look at it. They're going to break it down and then throw their trash out, and they will just keep trucking on. But not macrophages. Macrophages are going to eat it up, break it down, and then like basically come around and saying like, oh, hey, does anybody recognize this controller? And it'll just be going around like waving it around in a way. Does anybody know what this controller is? I found this. Does anybody know what it is? And eventually it'll come across a T cell, a T helper cell that might recognize, yeah, I know that controller. I've been waiting for that controller my whole life. And will come and interact with it, and that'll trigger its response further on down. That cannot happen unless somebody comes and presents that to the T cell. The T cell cannot find it. Somebody has to present it to it. That's self saying, hey, I'm self, and I found this non-self thing. Now we can go forward with all this. So it's part of the checkpoint system for them to be sure we're not, bombing ourselves, essentially. But yes, so our macrophages are going to be our in between here and our adaptive immune response. And our adaptive immune response is still going to help activate even more macrophage action because they are phagocytes. We want them to keep eating things up and breaking them down and killing them. So they're our big friends, they're our biggest friends, but neutrophils are going to be there sooner. They can respond faster. They're going to be able to come there immediately with those very first signals with those cytokines. So neutrophils, monocytes, and macrophages, which makes sense since macrophages were monocytes. They're our biggest phagocytes. There are other ones. All right, let's go through them. Neutrophils, they are going to come along pretty early on in the inflammatory response. They are the most numerous white blood cells in our bloodstream. Of all of them, it's like 55 to 65%, I think, of your blood, white blood cells are going to be neutrophils. They are the primary component of pus as well, because they're the first ones there at the site to eat up the bacteria. And the pus is going to be the dead bacteria, the live bacteria, some of it too, and the neutrophils, the fluid that came with them, all of that stuff mixed together. We also have eosinophils, which are more phils. And these guys are usually associated with parasitic infections, but yeah, I mean, they're also involved in allergies though. Like in the United States, that's typically what you're going to see them associated with is allergies. If not cancer, that's another thing they can be associated with. Monocytes and macrophages, again, macrophages came from the monocytes after they got the right signals and moved into the tissues. They will go and scavenge for any other foreign matter to present it to the lymphocytes to get that side of things going. So it is sort of like you would be having your scout, your mast cell or whatever at that site where you step on your nail, and then it pokes in and your scout says, uh-oh, we've got bacteria on the way. It poops out all of its signal, brings in the neutrophils right away. They start acting out and making more signal. Meantime, the macrophages might get brought in and pick up some of the waste that's being made, go and activate a T cell and start get them going. They're the messenger to tell the tanks and the bombers, we need to get cranking, but they're not ready to go right away. So in the meantime, the neutrophils are going to stay fighting and doing what they can.
T cells have, like you said, they were like waiting for this their whole life or something. So where do we get specific, like we've been exposed to that before, we've had a vaccine for that, or like how do they know?
They literally were. So yes. So how did they get the idea to be anti that thing?
Yeah, right.
Yeah, yeah.
Are they so specific?
They're that specific because your body made them that way. Okay. So your body, whenever it's going through making your antibodies and your T cell receptors, which are kind of the similar functions but on different cells, right? They recombine your genes. So like what? Because you don't have enough genes to make up for all of the things you could possibly interact with. So what happens is while they're maturing in that bone marrow situation, they are literally rearranging their genes to make up as many combinations as possible. They will also remove and insert, nucleotides and stuff just to give more variability to it. So yeah, they will recombine. There's like a set number of genes that you have in your body for like what we call the V, D, and J segments of these guys. There's like 3 segments, and you have like a whole bunch for each one. And it has to take at least one, and it can recombine with this one, and that one can go with this one, and whatever, and insert guys in between or delete guys in between to get more variability. So it is all depending on, because they're antibodies and the T cell receptors are proteins, that folding that they're going to get as a result of that variability. And so your body will do it with it that way. So you only have 20,000 genes, but you can make antibodies and T cells for like 8.5 billion different antigens. It's very impressive. Like scientists, how do they study that? People ask me these questions. I don't know all the answers to those stuff because I didn't really study a lot of like how they did their research with this, but usually most of it, honestly, it's going to start off with animal studies, of course, and looking at their immune system and their immune responses. And then, of course, that most of this probably couldn't have happened without PCR and our ability to look at genetics because of that. So it's just a combination of those sorts of tools. But that was a good question. But yes, so this is all starting to come together. We're starting to see why our innate response is so important before our acquired, but why the acquired is just like absolutely must have it happen. Because our innate response with our neutrophils and everything, that's great and it's doing stuff, but you don't get memory from that. So these guys that keep infecting you can keep infecting you unless you have memory built up, and those bombers are always ready to fight that specific thing. That can't happen without this part first. So our phagocytes that are going to be playing their biggest part in all of this, breaking down the bad guys, and the macrophages then going and presenting the pieces to our T cells to get them activated, and our T cells being our like regulators of all of that. That's phagocytosis. Chemotaxis is being called to that area by chemicals. We talked about this with bacteria, yeah? Same thing as like when I had the cookies up here and they smelled nice and you guys would want to move towards the cookies or away from poop. Those chemicals that we're smelling, they are getting the signals from their cytokines. Then they will come to the area. They'll recognize these guys with a PAMP. They'll come and bind on and figure it out and be like, oh, I know this. This is peptidoglycan. Okay, so we're eating this up and digesting it because I know this is PAMP. I recognize this PAMP. All right, so it would break that down in ingestion. So we, as a phagocyte, when you do that, you're literally engulfing it into a vesicle. That vesicle is called a phagosome. So a zome means vesicle, that's all. It can have stuff in it or whatever, not. In this case, it just has the thing you ate up, the controller. So we phagocytosed the controller into the phagosome. Then there's other vesicles in the cell that are going to contain toxic materials that we're going to merge with it to kill the stuff in there. So we literally have vesicles just waiting in these phagocytes that are filled with toxic chemicals like lysozyme. Remember how that's going to break down our peptidoglycan and our toxic oxygen molecules and acid and things like this, a whole bunch of stuff. Let's just load up all the grenades in here and like engulf it and just wait to pull the freaking pin out of that grenade when it merges in with the bad guy. So that's what happens. That phagosome and that lysosome with all the toxins are going to merge together and create the phagolysosome. And then it's like jumping a body into a vat of sulfuric acid or something. This is a really violent lecture. And and it's going to take a while, but it will definitely break it down. It takes about 30 minutes to kill bacteria in the phagolysosome. Then so that's the destruction process. So we've destroyed the thing. And now we will excrete whatever is left over. We don't want it anymore, especially if we're a neutrophil. If we aren't a neutrophil, if we're a macrophage, we might be sticking some of it out there being like, does anybody recognize this? Instead of just getting rid of it. But not all of them have the ability to do that. The neutrophils can't do that. So we already went through all this stuff. Again, we're going to recognize based on those PAMPs. And we already talked about what some examples would be. The phagolysosome happens when the phagosome, the vesicle we did the eating with, merges with the lysosome with the toxins in it. And that creates the phagolysosome where we're going to break things down, digest them, and then we're going to excrete or waste. When we get into talking about inflammation, We have some Latin-based terms that will probably make more sense to you, those of you if you speak Spanish at all. And I wish that I had taken Spanish. Honestly, genuinely wish I'd taken Spanish instead of German when I was in undergrad, because it's useless. Okay, but that's what I decided was, you know, my vibe. So whatever. But rubor means like a ruby, redness. It's usually from that vasodilation in that area, so increased circulation. That's redness from your blood underneath the skin. The calor is the warmth. So you touch somebody that has an inflamed area, they've injured even like deeper tissue injury or something, you can still feel the warmth that's going to be in that area. That's from the blood flow to that area. That's going to make it warm. Calor, if you think of like calories, literally a calorie is like a measure of like burning like a carbohydrate or something. I don't know what it is. You have to ask Dr. Tim. I don't remember what exactly it is, but you burn it and how much heat comes off of it. That measure of heat is called a calorie. So that's where that's where the word calorie comes from, is calor. It says warmth. Tumor obviously would be swelling from the fluids getting into the areas. If the neutrophils are coming there to fight the good fight, they came with fluid. That's just how it goes. They always do. And that's going to be that that plasma portion of your blood, the clear portion. All right, the dolor is the pain. Cool. You can also have loss of function. Won't always see that, but you can sometimes see that associated with the inflammation. When you have chronic inflammation that's going on for very, very long periods of time, you can see it presenting as another kind of disease if that is, left to go long enough. You might not even realize it's related to the inflammation. All right, so why do we have inflammation? What is the purpose? Why do we have all this rubor and calor and dolor and tumor? Like, why are they there? Well, it turns out that with inflammation, it will help mobilize and attract immune components to that site. Getting blood flow to that area and the fluid to that area, obviously, that's going to help with that. The redness and the swelling and stuff just happen to come along with that as we're doing it. We have stuff in the fluid that's coming in with it, like our fibrinogen, things that we're going to create clots with. We need that to get there. So that's our repair process that's going to start happening. And then we're going to clear away any of the harmful substances, any waste that's from this bacteria or any toxins that they could be producing, including the fluid itself that's being brought in. That extracellular fluid and that edema, the exudate that's building up in there, helps to basically dilute out any toxins that are in that area. And then we can actually destroy microbes and block their further invasion by creating the clots or by digesting them with like our neutrophils coming in and everything like that. So these are the steps involved in all of these. The upper left, we have step number 1, stepped on your nail, mast cells see it with their binoculars and they're freaking out. So there will be vasoconstriction at that point because of what the mast cells are doing. Why vasoconstriction? Because it's trying to wall off that infection until somebody can get there to help. It doesn't want it to get into the bloodstream. So once the neutrophils show up, now we can start the vasodilation, bringing more blood in, because the neutrophils are like, no, it's good. Now start the blood flow in, get everybody moving in here faster. We're going to get this. We're going to take care of it. And plus all of our clot forming factors and things like this are going to come with it. So we can start sealing off all of this and destroying these bacteria. So the neutrophils are going to continue to eat up and destroy the bacteria in the area. That neutrophils, bacteria, everything that's dead in there, as well as the fluid that came into the area, that's our pus. And then as that's continuing to go fight on, and we had macrophages that will come in and recognize some of this stuff and go activate the appropriate lymphocytes. And once the bombers and the tanks get there, the fight's over. We're done. Like, we're good. They take care of everything pretty quickly. So quickly, like we just said, that once they start making what they're supposed to and making antibodies and start attacking like they're supposed to, once they're ready to go, you can go along and see somebody that has the measles and not even know that you ever came in contact with the measles, because that stuff is so fast at acting. It takes a while to get there the first time. First time takes a while. That's what vaccines are for, to help us get past that. So we don't have to wait on it in natural situations. All right. Early inflammatory events, we of course have vasoconstriction and vasodilation. I'm not going to go over this stuff too much. This is stuff that I'm not quite as interested in, but yeah, you'll have clotting. clotting factors, platelets coming in and creating the actual, scabs and clots and things like this. Next is the edema. Like I said, this is leaking our fluid into the extracellular space. So outside of the blood vessels, as we're getting that fluid into whatever area it is that you stepped on with that nail or tack or whatever, that fluid is moving in there as we're moving the neutrophils in. It's also bringing in those clotting factors. That's exudate. So it's outside of the cells and it's moved out of the bloodstream to go deal with that. As the swelling is happening, even if it's in just small amounts, like you do stub your toe and it is swelling up and you can feel that there's firmness developing in that area from the swelling, that's edema, but it can of course happen on much larger scales than that. But edema results from exudate accumulation. And then how do we get rid of that edema? Well, that fluid, that exudate that formed the edema, that built up into becoming that edema, will have to get back into the lymphatic system, not the blood, right? The lymphatic system first, and then eventually, once it's gone through all of its filtration, go back into the bloodstream that way. And it will get reabsorbed, but that's how it's going to go. All right, so you guys heard me saying that these cells are going to squeeze in between the cells that make up your capillaries. In those very thin areas of the capillaries where you have like one cell thickness, they literally will squeeze in between the cells. That there's like a reason, the cell junction, I guess you could call it, or whatever, but they're going to squeeze through. So your white blood cells are capable of doing this. Your neutrophils, your macrophages and things like that can literally squeeze through there. That motion of moving from the bloodstream into the tissues by squeezing in between in the capillaries, that's called diapedesis. And that's a term you guys need to know. How do our white cells move into tissues from the bloodstream? Diapedesis. It just squeezes through. That might be one that you guys aren't able to say. That's okay. I forgive you. Yes, chemotaxis. We kind of already talked about this. I don't really know what else to say, but yes, all of this is happening thanks to signals bringing things to the area. So our chemokines or cytokines, the same kind of deal for our purposes. Calling those white blood cells to those areas. That's the chemotaxis that we're talking about. What do we need edema for? What do we need chemotaxis for? Like, why are we being called in here and then bringing all this fluid in there? Edema will be diluting things, bringing in the molecules that will help build clots, and the neutrophils are going to come with that due to the chemotaxis. They're called there by it. And that fluid with the neutrophils will help destruction of the pathogens, whatever they are. Then eventually our monocytes, our lymphocytes, and our macrophages will get involved. The macrophages will clear up all the pus and everything that the neutrophils are getting involved in, and also use any of that waste to present it to the lymphocytes to be sure they're getting activated. They will also get cross-activated during that. They'll basically, like, they go in and say, hey, I found this thing. We need the bombers in the tanks. And they're like, good job, little macrophage. Go on and do these things now for us. And they get more signal to go back and fight the good fight, essentially, and they will go and do that. They're very effective at that, actually. So cool. Then we'll replace that tissue with new tissue, form a scar, because your body doesn't care what you look like. Okay, so fever is part of this. We know if we're going to have calor, that warmth that's associated with all of this, that is part of it, right? That's the warmth. So why do we have fever? Does it itself have a purpose other than, it's just a side effect of this? And it's not, okay? It's not just a side effect. Fever is triggered by pyrogens, like a pyromaniac. Like a pyromaniac plays with fire. Fire is hot, fever, okay? So pyrogens can be exogenous or endogenous. Exogenous pyrogens come from like bacteria, pathogens, things like that from outside. Endogenous is whenever you have signals from your immune cells that are activating the fever. For whatever reason, they saw something and then they're releasing all these, cytokines saying fever, fever. So if it comes from our cells telling it to do it, that's endogenous. If it came from the existence of a fungi or a bacteria, that is exogenous. One of the most common ones, as it says down here with this endotoxin, is one of the most potent exogenous pyrogens that there is. It can lead to shock just by being in your bloodstream. So what does it do for us that's good? It inhibits multiplication of any of these microbes that are sensitive to temperature. It impedes them from getting the iron that they need to live. it slows down their enzymatic reactions because it inhibits the iron and also the temperature that would be ideal for them to have these things happening at. It's too warm. It increases our metabolism and helps speed up hematopoiesis and phagocytosis and the response of our lymphocytes as well, to be sure that we're getting as much out of our immune response as we possibly can. So fever has a purpose, it has benefits, and we want fever to a point, right? So if, for the most part, most experts would say they probably advocate allowing a fever to run its course in an infection unless it gets into that high range. We don't want that for us or for our children. And of course, if you have children, you don't just go by what I'm telling you, you go by what your doctor says. Or me, 104.5. I had it during COVID. Yeah. Yeah, that's really close. Yeah, I had to take an ice bath because it was like pretty early on, still like in the infection. Like it was like day 2 of. Yeah, I couldn't, like, it was like I felt like I didn't know what was going on. I was just confused. And so my husband like just dumped all of our entire freezer into the bathtub with some water. And he made me lay in there for like 20 minutes or something until my temperature started going down. And I was taking Tylenol. Like this was like with medication. So it was nuts, but that was terrifying. I had to take 2 ice baths because of that. The whole time I was sick. Yeah, fevers, man, they're rough. Yeah, don't play around with them. I mean, if you get to that like point, if you can get to 104.5 or something, and it's not showing any signs of abating when you do take medication or something, taking an ice bath is not a bad idea. But if it's still not getting better, then probably hospital is the better idea. Okay. If it's a kid, for sure, hospital. All right. So interferon, I like interferon. This is a good one because it's a good story to tell. It's more violence. Okay. Interferon is just another of these cytokines. It's a signaling molecule. It's a protein. And it's going to be made in response to viral infections specifically. So interferon, you get infected by Jim over here on the left. He gets infected by a virus. Okay, so the virus in this case, let's say it's a burglar that's coming into the house and ransacking everything. And so it gets in when he wasn't paying attention, and he's hiding out in his closet. Okay, Jim is. And he notices these burglars going through it, ruining everything. So he's like, I got to call Kenny. I got to warn him. So he calls Kenny, and that call is interferon. It's the warning call. Okay, so it's stuff made by that cell that's being infected to let the cells nearby know that viruses are coming, burglars are coming. Okay, so Kenny says, thanks for the warning, Jim. I'll have the shotgun ready. And he does. And so we have these antiviral proteins that we were able to make ahead of time to prevent the virus from taking hold in the cells. So we can actually give people interferon with certain infections to help boost that response and prevent more cells from getting infected by infusing people with that. Pretty cool stuff. Yeah, I know. Complement, this is the last of it. Complement cascade. It is a cascade reaction, and that's why you'll probably hear me always calling it the complement cascade, because it's like 50 proteins in your bloodstream, but like one of them will see something that it recognizes. There's 3 possible things, and we'll talk about what they are. But it'll see the thing that it recognizes, and then that causes it to trigger something else below, and then that triggers things below it, and then each one can like do more than one, and then it's just like multiplies. It's a big old cascade of a reaction once you see that one trigger by that one protein at the top. So 50 proteins that are always circulating in your blood. You do not have to wait on your body to start making these proteins. They're always there hanging out in your body, and they're going to help the inflammatory reaction. But this cascade response, one protein activates the next and the next and the next. These are the 3 things that can trigger it. They're called the classical pathway, the lectin pathway, or the alternative pathway. The classical pathway is triggered by the presence of antibody. If antibody was made by B cells and it's attached to something, then the complement pathway is activated, the classical pathway. If lectin is something that binds to sugars that we find on microbes. Okay, so if we see lectin binding onto those sugars, it activates that lectin pathway. It's the complement cascade. And then alternative pathway, you can have repeating molecules on microbes, a lot like the PAMPs. If it sees something very similar to PAMPs, and that includes LPS, it's the most common one that we could say, our endotoxin, lipopolysaccharide on our gram negatives. We see that, and we can recognize that directly. with our complement proteins, and that will trigger the alternative pathway. But they all end up doing the same thing. Whichever how way you got there, they all do the same thing ultimately. And this is how it will work. You don't need to know what C3 and all this crap is, okay? But that's just know that it's a cascade that's happening. They're going to involve one another. But once we start activating the pathway, we have two intermediate things that can come out of it. And that is inflammation on the left. We can help out with the inflammatory process, some of our intermediate proteins that get made. On the right side, we have phagocytosis, and I have pointed at it and said opsonization. Opsonization, or we say an opson, but this is essentially helping out phagocytosis so that it can be more effective. And what I mean is these complement proteins can attach to the surface of the bad guys to make them easier to eat up. So they'll go and stick onto the bad guys and make it easier to eat them up. And so we're helping phagocytosis. That process is called opsonization. We'll see it again in the next chapter. And then we have continued amplification and cascade leading to polymerization. So the things that we've made on the way down can come together and create a big old unit called the membrane attack complex, which does just what it sounds like. It's going to poke holes in membranes and cause the contents of those cells to leak out, and it kills them. It's pretty effective considering these are just proteins floating around in your bloodstream. So this is just kind of summarizing over that, but yeah, your initiation with your 1, 2, or 3, that's going to be your classical, lectin, or alternative. Your amplification and cascade in the middle in there, we can have our phagocytosis being helped via opsonization, and then the inflammation. Then we have the polymerization of all those cascade elements, leading to building the membrane attack complex, poking the holes, and leading to the death of the bad guys. Okay, that's it for innate immunity. Yes. Next time we will be back to doing our Kahoots and stuff beforehand. So we'll do the Kahoot next time over this chapter 15, and then we'll go into this like usual. Okay, let's go to lab. Get it over with. Let's see.